Okay, now I have started recording. So you can see my slide, right? So next, uh, like I started with why living system, but still you can say that you don't need to study living system in details to do what the scientist actually did. This because here you don't need to study about biology in details, right? This is just some kind of observation and some good effort, and that's all. I agree with you. And the next one is another example I will give it to you. So this is all of you are aware about today's kind of bullet train. So this is bullet train. So and I would like to tell you that mostly bullet train started in Japan. So the background was something like this. In Japan, a railway system, there are many tunnels inside the railway system, right? So when those trains are actually uh, entering into the tunnel, so as a result of them, that some air pressure built in just front of the train. And finally, when the train is just emerging from the tunnel, some air pressure differences happen, and that causes, a lot of huge sound. And that is kind of a problem. So then that sound you can hear from long distance also. So Japan railway system, they wanted to address this problem. So one of their railway engineer, Izzy Nakatsu, so around in 1980s, sometime during that. So his hobby was bird watching. So he found that this small bird, all of you know kingfisher, right? So this bird, when they go for fish and they hit the water, they don't disturb the water. They catch the fish without rippling the water. So as a result of that, he was thinking if we can design something like that, and that can solve this problem. So this is called like Shinkansen bullet train. You can forget the name, and whatever I'm telling you, the story kind of thing. Those are also I have already pasted in the notes section. So after the class, when I will send you the PowerPoint presentation, you can go through this short description what I am actually discussing now. But what he did, he redesigned the front part of this train, and he put here like almost 50-foot long steel body, like this nose that looks like this kingfisher's beak. Okay. And that way he solved the problem of building air pressure at the front of the train, and also uh, it improves the fuel efficiency. And this kind of bullet train can run even more than 350 km per hour, and in better track, in I would say magnetic levitation system, it can go more than 600 kilometer per hour speed. So that is really high speed, right? But this is from his observation. And also, again, you can tell me this is another good observation, but no need to understand biology in details. That is true. But this one, I'm going to discuss another example here where you have to understand biochemistry, what is happening inside the cell. So what happened? This is a small, animal organism. I would say like its name is the tardigrades. You can forget, no problem. Like you don't need to remember the name, tardigrade. It's kind of millimeter long cousin of arthropod like cockroach, right? It's very small only. But the beauty of this animal is when the environment is not suitable, no water, lot of problem, it can dry it up. It can dry it up for more than 100 years at normal temperature like 37 degrees centigrade, 40 degrees centigrade, no problem. It will be there. But when the weather is suitable, you have water, like comfortable, like suitable weather, this animal will start working again, right? So they can, like, see, this is called anhydrobiosis. So without water, somehow they can protect their information, what is present inside the cell. They can preserve their chemical thing inside the body so long, like years after years, more than 100 years. But now, if we can learn this thing, that will be huge help for us. Why? Because you will be surprised to know in many countries, particularly for poor countries, the vaccination is not because that we don't have vaccine. Forget about this COVID vaccine, because this is something different. Because no other vaccines we need to make in such mass scale. Like for example, if you think about child immunization program, right? Some specific vaccines need to be taken at different age. But still in poor country, in remote area, the problem is transporting those vaccines. In many rural area, remote area, not only in India, in many other part of the world, the problem is the transport, and even after transportation, how you will store it, because most of those things will be will be destroyed at room temperature. So now people across the world, mostly some pharmaceutical companies, they are trying to make or mimic this system, that whether we can make and preserve some useful drug, for example, vaccine, and without refrigeration, still we can do. So San Diego-based company Biometrica in United States, so they made similar kind of observing this thing, understanding this organism. They made some kind of devices so that you can put your stuff like precious biological sample, for example, vaccine, but you can store at room temperature, right? And another company in UK, they also did the same thing. So those are important. And in order to do that, you need to understand biology at this point, that how this organism preserve its chemical entity for so long time at room temperature, at even higher temperature, just outside. And when it gets suitable weather, it's just survive again. So that's a beautiful example. Now, I'm going into more like your zone, not mine. So that is current data use, right? Say I'm just taking this from Google, that 20 every minute of the day, that how much data getting used, and this kind of huge problem, the storing data and how it get used, all those things. So you can go through it later, but what I want to say here, I would like to summarize here. This one I started at the beginning. Some of you didn't join by that time. So digital DNA, I was asking about this, whether you are aware about it or not. Digital DNA. So Microsoft, they started this project in 2015, sometime during that. So, and also in some other university also, they are working on it. So the idea is whether you can store or whether you can use DNA as storage device, whether you can store information there. So in order to discuss that, I will just point out some of the important thing here. So this one is hard disk, as you can see, and then flash memory like flash drive, and this one is DNA. We just mentioned here bacterial DNA, but any DNA is fine. So now if you see, in case of DNA, most of the things are like advantageous. The only problem is the read and write speed. But if you see data retention, it's much better in DNA. Power usage, that is also good. Data density, much higher in DNA. But only thing is that how you code the information in DNA. And not only that, you wrote the information, then how you understand, how you decode that thing, right? So those are the some important things. And here you can see that what we are trying to say, this paper came, I believe, around 3 years back in Nature. Like I will send you the paper also. I will just upload in your MS Teams. So that weight of the DNA needed to store Wallace data around 1 kg of DNA. That's all. To store like Wallace data. So you can understand this is because of very high data density here. So now here I'm just giving you like showing you that Microsoft Corporation, their DNA storage research, and they try to. So only thing is here that I will discuss later also. In computer you just say 0101 binary kind of code, but here in case of DNA we have 4 different nucleotide ATGC. So you have some advantage. So here I'm not going through these details, but you will see later that here they are trying to explain that how they code information. Information means here they use like some line from Shakespeare's poem. Actually, see, it's written here. You can go through it details later. Okay, and this one is the last slide, like introductory slide, I would say. A computer scientist guide to cell biology. I would like to particularly mention here that Professor Cohen, so he's a professor in Carnegie Mellon University at United States, and his area of research is artificial intelligence and machine learning. But he is expert on that field. But what he did, so he understood a long time back that many people started working in the field of biology. They are trying to address, they are computer scientists, they are engineers, but they are trying to solve a problem. The problem rely in biology, the biological problem. But it will be better to solve the problem if you understand biology to some extent, right? So what he did, he even wrote a book, like a computer scientist guide to cell biology. I have the soft copy of this book. So the thing is, like this is really good for some, I would say, for I can tell this book something like that, biology for non-biologists. So the way he wrote the biology, so that it will be easy for easy to understand by other group of people, because sometimes biology is difficult because of just lot of different types of name and other things, as I started before, right? So I read his book also. So I'm like, you will see that in most of my classes, I will also try to, bring very minimal amount of complicated name and other things. But this is just, introduction. Now I will go into, next topic. But before that, I want to discuss with you. If you have any question, any suggestion, I'll be more than happy to discuss with you. You can unmute and you can talk. So here, the components of living systems. So all of you are aware that we have in our body, we have carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus that constitute the living system, and different types of a chemical entity like DNA, protein. They have different combination of this element, and there are 4 major groups of biological macromolecules. So proteins, carbohydrates, lipids, and nucleic acid. That you have to remember. all of you are aware, right? Lipid means fat. And carbohydrates, normally what we say sugar, that is example of carbohydrates. Protein and nucleic acid. So I will discuss in more details about proteins and nucleic acid. So now, today I'm going to mostly concentrate on nucleic acids. How is genetic information stored in our body and how it get maintained, right? So now, I would like to first discuss one experiment here. I would like to tell you, and then I will discuss more about the DNA. So for example, DNA is our genetic material. I think that since you are not talking, I'm just assuming that all of you are aware about it. But I told you that if you have any question, anything need to be discussed, just unmute yourself and stop me and discuss me, no problem. So I hope that all of you know that DNA is our genetic material. DNA is genetic material of living system. Only few viruses, for example, this coronavirus, they have RNA as their genetic material. But most of us, almost all living forms, they use DNA as their genetic material. But how you will prove that DNA is our genetic material? What is the experiment? So I'm going to explain the pioneering experiment done by Griffith. So scientist name is Griffith. So he did the experiment. He started the experiment, but he was unable to finish the experiment. But that actually proved that DNA is our genetic material. So if you see, today we are suffering because of this pandemic, COVID-19. Almost 100 years back, another pandemic, it was more devastating than what we are observing now. That was 1918. Do you know what's the pandemic? It's happened in 1918. Spanish flu. So Spanish flu. So millions of people died actually, approximately 100 million people died across the world. Considering the population during that time, it is huge number, right? So during that time, across the globe, many scientists, they were trying to discover vaccine, because not only because of the Spanish flu, many people get sick and then they are getting infected by another bacteria. This is called secondary infection. That means when you are sick with some viral, with some other disease, you might get quickly infected by some other, opportunistic bacteria, other bacteria, which are generally not very harmful, but when you are sick, then you might get, attacked by those pathogens. So that time, what is happening? Another bacteria that is called Streptococcus pneumoniae. Streptococcus pneumoniae. So this is the bacteria. So many people were suffering from pneumonia because of this infection just after the Spanish flu. And as I told you, that many scientists across the world, they were trying to make vaccine against this. So in 1928, Griffith actually did some experiment. So you don't need to remember this name here, nothing. But the experiment, you have to understand. That is very important. So what he did, I will explain, but before that, you need to know that the Streptococcus pneumoniae, they are bacteria, and there are two forms of bacteria, two different types of bacteria. One is kind of, you can say S strain of this bacteria, and the other one is R strain of bacteria. S stands for here smooth surface, and here R stands for rough surface, right? Smooth bacteria and rough surface bacteria. So if you just remember this, in addition to that, I would like to tell you that this smooth bacteria or S strain, they are virulent. Virulent means if you inject this bacteria to mouse, the mouse will get sick and they will die. So this is called virulent. That means this strain is lethal, right? They can cause the disease and they can kill the organism, virulent. And this is non-virulent. If you inject this strain, R strain, to mouse, just for experiment, mouse will not die. They will survive well. Why? Because of this layer. Whatever the layer, this smooth bacteria they have on their outer surface, that is causing problem. That causes problem. And that's why they are virulent. Now the thing is, what Griffith did, so he injected mouse, this is a small mouse, right? Injected R strain of bacteria. And tell me what will be the result. Just unmute yourself and tell me. So if you inject R strain of bacteria to this live mouse, what will happen?
It will not get sick.
Yeah, very good. So it will, so that's why, like, discuss, no problem. Okay. Because then I will, like, I cannot see you, you cannot see me. So if you discuss, that will, that will be good for you and good for me also. I will at least understand that at least one student can, answer this question so that you are listening. So the mouse will survive, right? No problem. Mouse is surviving well. Now, next experiment he did. A strain of bacteria. What will happen? Mouse will?
The mouse will die.
Yeah. Mouse will die because a strain is, virulent strain. Now, total 4 experiments he did. Mouse plus, this is very important. See, heat killed a strain. Heat killed a strain. So what do you think? What will happen?
The mouse will survive.
Mouse will survive. Very good. Heat kill means, this is in lab we say we autoclave it. Like you just, in at home you see pressure cooker, like almost similar temperature, similar pressure. So, you just sterilize it. So as a result of that, very high temperature, so heat kill. So bacteria, they are completely gone. That means they are dead now and mouse is surviving. Mouse is surviving. Last experiment, this is the most important experiment, what he did. Another mouse. So when we do experiment, I'm just like, showing you just one mouse, but we need to do this experiment in a group. Say in each group, we might have 5 mouse, right? 5 mice. So that is, we need to do, but for explanation, it's okay. So what he did, he killed S strain plus live R. Live R and heat killed S together, mixed together. And then he injected. What will be the result? What logically you think actually? Tell me that. You don't need to like provide the right answer or wrong answer. That doesn't matter. What you think? What should be your answer?
Mouse will survive.
Yeah. So if I am also in your place, I will say mouse will survive because it killed a strain and liver. Liver, they don't have effect, right? And it killed a strain already died, so mouse will survive. I would also say the same thing. But it's a huge surprise for Griffith and for all of us that all mouse, whoever he injected this group of it killed S and liver, mouse died. This is the experiment from where he pointed out that this heat killed S strain, this bacteria, although as an organism they are died, some information might be present in those dead cells. And those information are getting transferred to this live cell. And as a result of that, this live R strain, they are appearing as smooth strain also later. This part is completed. I'm going to discuss you in details. So first of all, when I discuss here that two different strain, S and R, why they are different? They are same bacteria. That means S strain, they have some additional information that is in the DNA, but at this time we don't know yet, but I'm just telling you that information is present in DNA, some additional information. They make this extra layer. This is some kind of polysaccharide layer, some kind of sugar layer, right? And because of this additional layer, our immune system, the immune system present in mouse, they cannot handle this S strain. So as a result of that, they are virulent. So what happened when Griffith observed that when he injected this group of mouse, it killed S and liver, mouse is dying. That means, as one of you answered that mouse will survive, I agree with that student that apparently it seems like so. But the thing is, what is happening here, that some information present here in the heat-killed cell, heat-killed S strain, they are getting transferred into this live R strain. And now, after acquiring that information, those R cells become S cells. And as a result of that, they can cause the disease and they can kill the mouse, right? But someone need to prove that. What is coming from the heat-killed S cells, right? Because what he told Griffith, that something that is called transforming principle. But what is that transforming principle? That was not clear by that time. Because unfortunately, this is a huge discovery. But Griffith didn't get Nobel Prize also and he didn't see or he didn't prove that this is DNA. He did up to this and he was carrying out some more experiment. But the problem is in 1941, you know that time is World War II, right? So during that time, some German bomb actually, destroyed his lab, and he was also in lab. He died in the laboratory itself. So that's why that experiment, was incomplete, cannot be done by him. But some other group of scientists, they actually did some additional experiment, and they proved that this is DNA. DNA is the genetic material. Why genetic material? That means the DNA from this heat-killed S strain transferred into the R cell, and R cell acquired this new genetic information, and they are making the polysaccharide layer, and they are causing death of this mouse. Okay, and in the next experiment, I will discuss how they prove that this is DNA. So this experiment that proves that DNA is our genetic material. So don't try to read everything. I will discuss very slowly whatever I'm doing now. And it will be better for you if you go with me step by step. So then the hypothesis here is the genetic material of the cell is either protein, nucleic acid. protein, DNA or RNA. So that is kind of hypothesis. Based on that, they designed some experiment. What is the experiment? So they took heat-killed S cells, heat-killed smooth strain of bacteria, the same bacteria, Streptococcus pneumoniae. And then they grind that bacteria. You make a suspension of that bacteria. So now, if you take those solution of that heat-killed S cells, and then you can quickly remove lipid and sugar by very simple biochemical method. And then in the tube, what are the major biological components remaining? That is nucleic acid and protein in this tube, because lipid and sugar is already out, right? And now what happened? They distributed this material into 3 different tubes: tube 1, tube 2, and tube 3. And in this tube, they add one enzyme. This is called proteinase. I will tell you how to remember this name. No need to, you can remember all these things. Proteinase. So in enzyme, all biological enzymes, all enzymes in living system, we put ASE at the end, ASE. So if some enzyme name is proteinase, that means this enzyme will cleave or will chop up protein. It will cleave protein. So proteinase will destroy protein. Okay. So in the first tube, they took the sample and they add proteinase. That means in this tube, no protein there. And that solution they put in a conical flux, and then they add the R strain of bacteria here, R strain of bacteria. So what they found that here also in the conical flux, S cell appears. So what will be your conclusion? That means that protein is not involved in transferring this information. Otherwise, this R cell should not be converted into S cells, right? Because here you don't have cells. You already grind those cells. You are just taking the solution and you destroy the protein present in tube 1. And in tube 2, they added, the multiple scientists did, that's why I'm saying that they added RNase, another enzyme that destroy RNA, ribonucleic acid. RNA and DNA, 2 major forms of nucleic acids. So when they add RNase, that means no RNA in the solution. Then similarly, they added R cells, R strain of bacteria. Again, they observed that S cells appear. That means RNA is not involved in transferring this information. In the third tube, they added DNase, enzyme that destroy DNA. So no DNA in this solution, right? And during this time, when they added R strain, those cells remain as R strain. No S cells appear. That clearly proved that DNA contains information. Okay? DNA is our genetic information, and DNA, like whatever Griffith observed, and if you combine both of this experiment, you can explain that DNA is our genetic material. And now all of us know, but I just discussed the overall experiment. So if you have any question, any discussion, you can continue. So everything clear? Or you are not listening?
Yes.
So are you aware about this experiment before?
No, sir.
Okay, fine, that's fine actually. But this is simple experiment, but that is very good experiment. Very, I would say, this is one of the beautiful experiment in like modern biology, I would say, in molecular biology or modern biology. This is very important experiment that prove that DNA is our genetic material, because you can understand that information, particularly this molecular biology, is very important for in the biomedical field also, because of many genetic diseases and so many things. In order to address those things, like you need to know more about genetics, more about the information, and particularly how those informations are stored, maintained, and how you can modify the information if required, right? So all those things are really important. So today, I'm not going to like continue the discussion. Like just I discuss about this experiment, and mostly like from next week, I will discuss more about DNA. That means how you can synthesize DNA and all those things. Let me check that what's there. Just next slide. Maybe another 10 minutes I will discuss. Then. Yeah, so here, as I told you that nucleic acid, we have two defined forms. One is RNA and DNA. So if I ask you, like, what is the difference between RNA and DNA? One of you can unmute yourself and you can try to answer that. What is the difference between DNA and RNA? You studied in your class 10.
Sir, one single stranded and others double stranded.
Okay, very good. Like most of the time, students say that. Like I'm happy that you unmute yourself and you like provided this answer. So yes, RNA is single stranded, DNA is double stranded. So this is like also like I would like to mention that at physiological condition in our body, many RNA can be can be there, like they can be there as double stranded form also, right? So this is not a huge difference, but although when I was also a student like you, when I studied biology in school level, I also used to study that this is the first difference, that RNA is single stranded and DNA is double stranded. But in my opinion, like or at this level, I would like to mention that that is not the major difference, right? Because there are examples when DNA is single stranded or RNA is double stranded. So that is possible. Okay. So some other difference, if like you can mention any one of you.
Sugar is ribose in RNA and deoxyribonucleic.
Yeah, exactly. So that is the huge difference. That's sugar. That makes everything, like why RNA is RNA and why DNA is DNA. That is coming from the sugar itself. So in DNA, nucleic acid is a polymer. Okay. This is a polymer. Polymer of what? The monomeric units are nucleotides. Nucleotides. That means nucleic acids are polymers of nucleotide. Now, the nucleotide, that means the monomeric unit, they composed of three groups. One is sugar plus nitrogenous base. It's kind of complicated structure. I'm not going into details. And then the phosphate. Okay. So these three moieties together, they form nucleotide. And the repeating subunit of nucleotides that makes polynucleotide chain, and finally RNA and DNA. But where is the difference? So this RNA, DNA, they contain sugar that is called pentose sugar. All of you know pentose means they have 5 carbon like in the ring, pentose sugar. And now if you see here, so this pentose sugar, in case of ribonucleic acid, they have ribose, and in deoxyribonucleic acid, they have deoxyribose. So one oxygen is less in this sugar. I will show you particularly with the structure where it's missing and what it does actually. That is the major difference. Now, here I have one more slide, that flow of genetic information or the central dogma of molecular biology. This is very important. You should understand this thing. So DNA goes to DNA. That means you have to make DNA out of DNA. Otherwise, see, I told you at the beginning of the class that we have 10 to the power 13 cells in our body. Although approximate, but like 10 to the power 13 cells. But all of us, we are becoming adult. We have 10 to the power 13 cells, but all started from just single cell, just one cell. And then now we have 10 to the power 13 cells. Like this is just single cell. Just after fertilization, we have zygote. And from the zygote, we have the adult human individual. So that means each round of cell division, that means from one cell to two cells, then 2 to 4, that way cells are getting divided. Each round of cell division, during that time, you have to replicate DNA. That means you have to copy your information. See, cells without information, no meaning. You have to have the information first. Otherwise, what will be the function? I told you that everything is written in DNA, the information present in DNA. So as a result of that, the prerequisite for cell division, that means from one cell you are getting two cells. But before that, you have to copy the information. That means whatever the DNA you have in that cell, you have to make double. That process is called replication. So DNA get double, and then that DNA will be segregated equally into the two cells. How it will be segregated equally, we will discuss in a different class. But the thing is that DNA should be equally divided into two cells. But the process of this DNA synthesis is called replication. It's happening very systematically in our body and very perfectly without mistake. You understand, right? If a lot of mistakes happen, then what will happen? We have wrong information that will make wrong product and that will create problem, right? So this is a very precise system where mistakes are almost negligible, no mistakes. Then the next step is from DNA to RNA formation. This is called transcription. This process is called transcription. Why transcription? Because DNA is also the polymer of nucleotides. RNA is also the polymer of nucleotides. I just mentioned what's the difference between DNA and RNA. Some sugar group, that is the major difference. But otherwise, they are containing the similar kind of, information, similar style, everything. They're just kind of transcript kind of thing. And the process is called transcription. And RNA, as one student at the beginning answered, that single stranded, you can see we are showing just single strand here. And finally, you have to make the product. Product means here protein. So what I want to say here, everything in a living system in our body, they are protein or they are a result of some other protein. Again, I'm telling you, everything in a living system, either that particular thing is a protein or that is a result of some other protein. So enzymes, they are protein. But to make all those protein, these proteins are called machine. But what kind of machine? Molecular machine. They are small, right? Very small, molecular machine. So in living system, they do function. They are the functional molecule. But how you will make your protein? What kind of protein will be, will be formed at a given point of time? So all those things are like finely regulated in our body, and the information present in DNA. That means what kind of protein will be generated or will be formed, how it looks like, what will be chemical nature, all those informations are present in DNA. I will concentrate within coming few lectures about these steps, particularly how this information gets processed. So I'm telling you that DNA is strictly informational. DNA contains the information. Okay? They don't function, they are not directly working in our body. Rather, they are storing the information and you have to process the information only. And this RNA is intermediate in between this DNA and protein. So RNA, they are called operational molecule. DNA is strictly informational and RNA is operational. RNA is intermediate between DNA and protein, and finally, protein are the molecules which will function. So this step is called translation. Why translation? You know what is translation in English language. Translation means, for example, all of you know, right? Just to say, if you go to, for example, France, right? So you don't know French language, right? So what you need a translator, like who will understand both French as well as your language, whether it's English, Hindi, or whatever regional language, but your language. So that is translation. So here also the same thing happening. See, DNA is polymer of nucleotides. RNA is polymer of nucleotides. Suddenly the language is completely changed. No chemical identity here. Protein is polymer of amino acids. Completely changed the language. So that's why this step is called translation. Don't forget, replication means to make DNA out of DNA. Transcription means from DNA to RNA. That's an intermediate molecule that helps to make protein. And finally, via translation, we get protein, right? And here I will discuss in the next class. As I told that today I will complete soon. Okay, so this is first class, right? Just started. So next week I will mostly discuss about the physical and chemical properties